Hi, this is Professor Johnson, and in this video we will be talking about control of the cell cycle and cancer. So cell cycle is important, or control of the cell cycle is important because you need to make sure that everything has been copied and separated appropriately before you divide into two different daughter cells. So when things go wrong with the cell cycle, that's when you end up with things like cancer. So it's very important that you keep the cell cycle under proper control, otherwise you'll have this uncontrolled cell growth that you see in cancer. So control of the cell cycle happens because there are certain points in the cell cycle that are irreversible. The first point that is irreversible comes during DNA synthesis. So once you've started making your DNA, once you started copying the chromosomes, you can't go back. Anywhere in G1 you can be building these things and preparing to divide, but you haven't committed to division. Once you've started synthesizing the DNA, you're committed. You have to continue all the way through the cell cycle. The second point that's essentially irreversible is when you actually separate the chromosomes. So there's this point in mitosis where you have your copied chromosomes, your two sister chromatids that have been copied, that get separated. Once you have separated these and started moving them apart, you can't go back. You're stuck there. So the cell has several checkpoints that it uses in order to make sure everything's in tip-top shape before it actually divides. The first one you might expect would occur before the synthesis of DNA. So it's after G1 but before S, or really as those two mesh together. So this is known as the G1S checkpoint. The second checkpoint is the G2M checkpoint. So it comes after the second gap phase, but before mitosis is actually begin. And the third one actually happens during mitosis. It's known as the spindle checkpoint. It's when the chromosomes are lined up at the metaphase plate before they pull away at anaphase. So what's going on at each of these checkpoints? Well, at the first checkpoint the first checkpoint is where things such as growth factors can act. So growth factors are going to be signaling molecules that can bind to receptors and signal the cell to start dividing, to say, hey, we're ready to divide, start dividing. Other things that are being checked for at this checkpoint, besides the presence of these growth factors, are the nutritional state of the cell. So is the cell healthy enough? Does it have enough things that it can successfully divide and copy its DNA? And finally, it's checking for the size. Is the cell big enough to divide? At this second checkpoint, at the G2M checkpoint, what the cell is checking for is whether replication has been completed. So have you copied all the chromosomes successfully? And has this replication of chromosomes occurred without damage or mistakes? So if the cell notices that replication has not been completed or that the DNA has been damaged in some way, it's going to stop the cell from dividing, go back and fix these, or if it can't fix them, it's going to go through apoptosis to prevent the cell from dividing and causing problems. Finally, at the spindle checkpoint, you're checking to make sure that the chromosomes are indeed attached at the metaphase plate and are ready to separate. So what happens when something goes wrong at these checkpoints is that the cell has the potential then to become cancerous. Now, a lot of things have to go right for cancer to occur. It's going to be simplified in this version of the video. So first, at the first checkpoint, one of the things can happen is you can have the growth factors be turned on without the growth factor actually being there. So it's telling the cell, hey, I want you to divide, even though the signal to actually divide isn't actually present. So you can think of this as being sort of a green light here, even though normally there's a stop sign. Now, these genes that do this, they function as proto-oncogenes. Proto means before, Oncogenes mean a gene that cause cancer. So these are genes that, when they're not mutated, are functioning normally to promote the cell to divide. But when they are mutated, they'll be telling the cell to divide even when the signal to divide is not actually there. So when they're mutated in that way, they're known as oncogenes. So oncogenes are genes that actually cause cancer. Now there's another type of gene in our cells that is going to regulate the cell cycle, and these are the tumor suppressor genes. So these are genes that are functioning sort of like the stop signs that are checking to make sure that everything's okay before it progresses. So it's checking, looking both ways to see no cars are coming before it crosses the street. This is looking both ways to make sure everything's right before the cell divides. Now, something to remember is that because all of our chromosomes come in pairs, we will have 2 copies of every gene within our bodies. So with tumor suppressor genes, you have 2 copies of these, and with proto-oncogenes, you have 2 copies of these. Now there's a difference in how they can cause problems, and that is best explained using a car analogy. So a car normally functions, it has an accelerator and it has brakes, and it has 2 brakes. It has a parking brake and it has a regular brake. So normal function, the car can start and stop as it wants to. So it starts, stops, doesn't run into the brick wall because it has control of the brakes, it has control of the accelerator, so you're good. Oncogenes, when they're mutated, is essentially mutating it so that the car always has the gas down. So you can imagine, whereas the car was starting and stopping before, now the car, whenever you let off the brakes, it's going to drive forward. You still have the brakes there, so you're not crashing into the wall yet. The brakes are still present, so you still have control over it. Now the brakes are the tumor suppressor genes. Now remember, you have 2 of these. What happens when you mutate 2 of the tumor suppressor gene, or 1 of the tumor suppressor genes? Well, when you do that, you still have function and control of the car because you still have the other brake to protect you. So you're still not running into the brick wall. What happens if you have both of these mutated? Well, you have the first one and then you have the second one. So what happens now is that the car runs into the brick wall. Notice that it was not speeding towards the brick wall, so it's not slamming into it like is going to happen when you really have problems, but there might be a little bit of damage going on. And of course, the car always has the option when the brakes are not working, just don't put your foot on the gas and you won't run into the brick wall. Now what happens with cancer is you have mutated both of the brakes, and there's been a mutation such that for some reason the gas is, or you're always accelerating. So perhaps the mutation stuck down the gas pedal so that it's always going forward, or the mutation, you mutated a nitro booster or something on your car that drives it. So you have 2 genes that could potentially do this, but you only need 1 to be mutated in order to drive forward. And when that happens, the car runs into the brick wall, you have the parts flying everywhere, and you end up with cancer. All right, I hope that video was informative to you. I will see you in class.